Have you ever wondered if it's actually possible to clone a plant? And I don't mean in some sci-fi movie, I mean right here, right now in a lab. What if I told you that you could grow an entire forest or a field of totally disease-free crops just from a tiny little piece of a single leaf? It sounds like magic, right? Well, it's very real science. So let's dive into this amazing world. So here it is, the big question. How can you actually do it? How do you take one little part of one plant and turn it into thousands, or heck, even millions of perfect identical copies? Well, the answer is a technique that's already changing agriculture and conservation as we speak. You do it with this. This is plant tissue culture. And what it is essentially is growing plants in vitro, that means in glass, under perfectly sterile, perfectly controlled conditions. You got to think of each one of these little jars as a miniature, high-tech greenhouse. It's got everything a tiny plant needs to just thrive. It's literally a whole ecosystem, all in one little jar. But what's the secret sauce here? What's the scientific principle that makes all of this even possible? Well, it all comes down to this one incredible biological concept, a single word, really, that explains how a small clump of cells knows how to become a whole plant. And that word is totipotency. It's this amazing built-in ability that's locked inside basically every single plant cell. It lets it access its full genetic blueprint and regenerate a complete organism. I'm talking roots, stem, leaves, the whole shebang. You see, unlike most animal cells, a plant cell never really forgets how to build the entire plant from scratch. And look, the research on this is absolutely clear. The entire technique, the whole field of tissue culture, is built on this one powerful idea. It's not some lucky side effect. It's the fundamental science that lets us tap into a plant's incredible power to regenerate. Okay, so if totipotency is the magic word, how do we actually perform the trick? Well, like any good recipe, you need the right ingredients and you need a clear set of instructions. So let's break down the step-by-step process for making these plants grow in a lab. So this process, it's called micropropagation, and we can break it down into four main stages. First, you get your explant. That's the tiny piece of tissue you take from a healthy mother plant. Second, you have inoculation, which is just a fancy way of saying you place that explant onto a sterile, nutrient-packed gel. Third, you incubate it in a special room with controlled light and temperature. And finally, the fun part, you just watch it develop into a brand new plant. Now, the real heart of this whole thing is the recipe for that culture medium. It's this super precisely made gel, and it's full of building blocks like macronutrients and vitamins. It's got a carbon source, usually sugar, to give the cells energy to grow. But the real secret ingredient, the game changer, is the plant growth regulators. These are basically hormones that act like little traffic signals, telling the cells exactly what to do. Hey, you, form a root, or you, grow a shoot, or just everybody multiply. So what happens when you master this lab recipe? What can you actually do with it? Well, the results are, honestly, staggering. This isn't just some neat lab trick. It's a technology that is having a massive impact on the world right outside the test tube. And this right here just shows the transformation perfectly. On the left, you've got what's called a callus. It's just an unorganized blob of dividing plant cells. But you give it the right hormonal signals, and that simple clump of cells can be triggered to grow into the thousands of identical, healthy little plantlets you see on the right. It is literally a production line for plants. So why should you care? Because the applications are huge. We're talking clonal propagation, making millions of identical, high-yielding coffee trees, for example. We can eliminate diseases. This is how the entire global banana industry actually survives, by creating clean starter plants. It's also a massive tool for conservation, saving endangered species like rare orchids from going extinct. We can use it for genetic improvement and even to produce valuable things called secondary metabolites. Think about the anti-cancer drug Taxol, which comes from the yew tree. We can make it in a lab without ever having to touch the tree itself. And don't for a second think this is some small, niche technology. The scale is already immense. In developed countries alone, we're talking about an estimated 212.5 million plants being produced this way every single year. A huge chunk of those are ornamental plants. So yeah, the next time you see one of those perfect orchids in the grocery store, there's a really good chance its life didn't start in a greenhouse, but right here in a sterile little jar. And that kind of scale, well, it really points to what's next. Because as we look to the future, plant tissue culture isn't just about making prettier flowers. It's about taking on some of the biggest challenges our planet is facing. The promise is enormous, right? We're looking at a tool that can help with food security, rapidly clone crops that can handle climate change, and conserve biodiversity. But, it's not a magic bullet. The process can be pretty expensive. There's always a slight risk of weird genetic changes happening in the cultures. And just keeping everything perfectly sterile, that is a constant battle for the people in the lab. So, all of this really leaves us with a pretty fascinating question to think about. As our population grows and our planet keeps changing, are we going to be turning more and more to these high tech greenhouses in a jar? Could it be that the future of our most important foods, and maybe even our forests, is actually growing right here?